after the successful completion of the 11th chapter. Let me start with the chapter number 12, that is the mineral nutrition. It was in 1860, Julius van Sachs, of a German botanist. He proposed a method called hydroponics. Hydroponics means soil-less culture. Ever since, number of improvised methods have been employed for the soil-less culture, and this kind of soil-less culture, hydroponics, is employed for the commercial crops like even the tomato, seedless cucumber and lettuce. So there are about 105 elements. Of these, the elements which are present in the plants are only the 60 elements. Sometimes even the selenium, gold, strontium are also present in the plant body. So there are some methods employed by the recent knowledge from the various fields to detect even the element which is low concentration of 10 inverse 8 G per ml. So essentiality of any element depends upon it should support the growth, it should play a direct role, and the deficiency of one element should not be met by another element. These are some of the essentiality, shows the essentiality of the elements. But the essential elements should they are present in as a structural component and the energy related reactions, and it should activate or inhibit certain enzymes, and it should alter the osmotic potential. Say for example K plus and Na plus. So these things they alter the osmotic potential in plants. There are two kinds of nutrients, macronutrients and micronutrients. Macronutrients are the elements which are essentially present in the plants. Their concentration is more than 10 millimole. That is the macronutrients. The important macronutrients are CHO, NPK, MSC, code word, carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, magnesium. S stands for sulfur, not for sodium. This is very important. This is the sulfur, calcium are the very important macronutrients which are occur in the plants. It is more than 10 millimole, and there are certain elements which are needed in less amounts. That is called trace elements or micronutrients. There are about 17 trace elements. So boron, the chlorine, copper, manganese, molybdenum, iron, sulfur, zinc, nickel, and even the sodium, silicon, cobalt, selenium. There are about 17 micronutrients which are needed by the plants, less than 10 millimoles kg inverse, inverse. And then coming to the first one, CHO, carbon, hydrogen, oxygen. They are observed by the plants and present in the salts absorbed by the plants, and carbon, whether it is carbon, hydrogen, oxygen, they are present in carbohydrates, proteins, lipids, and play an important role in the growth and other metabolisms. And coming to the nitrogen, that is very important. Nitrogen absorbed by the plants in the form of nitrate and even present in the form of nitrite, ammonia, etc. And nitrogen not only present in the carbohydrates, proteins, lipids, but also in the nucleic acids as nitrogen bases, and even the proteins, that is the amino acids, and even present in some hormones also, and it is also present in the chlorophyll and the vitamins also. But very important thing is nitrogen present in the meristematic tissues. That is also very, very important as far as the nitrogen is concerned. Coming to the potassium. Potassium activates enzymes and maintains the osmotic concentration. That is the water and the salt balance along with the sodium. And coming to the calcium as well as potassium and nitrogen, the KCN, potassium, code word KCN, potassium, calcium and nitrogen, they are present in the meristematic tissues. As we know very well, the calcium also present in the middle lamellae as the calcium pectate, and calcium plays an important role. That is also it is required for the spindle formation. Apart from this, the magnesium activates PEP carboxylase and RUDP carboxylase, and it is the non-transitional metal which is very important, non-transitional metal which is present in the chlorophyll, and magnesium is required for binding the subunits of the ribosomes. And magnesium is the metal which is present in the chlorophyll, as I told you earlier, and magnesium also plays an important role in DNA and RNA synthesis. That is very important. Next one is the sulfur. Sulfur is a macronutrient, and it is present in the coenzymes, vitamins like the biotin, thiamine, vitamins, hormones, even ferredoxin and ferredoxin reducing substance, especially the coenzyme A. And the important thing is sulfur also present in the amino acids like cysteine and methionine. So iron, it is present in the cytochromes FD, ferredoxin and the ferredoxin reducing substance and constituent proteins, and it activates catalase, which is very important for essential for the chlorophyll synthesis. Next to this, iron, zinc activates the pyruvic carboxylase as well as the pyruvic carboxylase, alcoholic dehydrogenase, and it is the zinc which is present in auxins. This is very important. So this chapter is very, very important for the NEET examination. So because for this chapter you require the knowledge of photosynthesis, respiration and various growth and other activities of various activities that takes place in the plant body. One should have the clear knowledge about all the physiology, then this chapter becomes easy, or else you have to remember line by line. That is very, very important. And coming to the boron helps in the calcium uptake, and it is the boron which helps in the pollen tube germination. As you might have observed in the practical, some teachers, they use boric acid to promote the pollen tube germination. That is boron. That is also very important, and boron is required for the root formation, and very importantly, this boron helps in the transportation of sucrose. So, as we know very well, the sieve tube elements are the rectangular structure, contains sieve plates, and cytoplasm chiefly contains sucrose and P proteins, and sugar alcohols like mannitol, sorbitol, also P proteins, that is phloem proteins. And this sucrose transportation, it requires boron, which is very important. And coming to the copper, it activates enzymes and also regulates reduction and oxidation reactions. And coming to the chlorine is very important. Anion cation balance it maintains, and chlorine plays an important role in the photoionization of water, wherein that is evolution of oxygen, in which also it plays an important role. and the 4 electrons are the electrons produced because of the photoionization of water return to the chlorophyll during non-cyclic photophosphorylation through chlorine. That is very, very important. And molybdenum plays an important role in nitrogen fixation, and molybdenum and iron they are the components of nitrogenase enzyme, which converts the different forms of nitrogen into ammonia. That is very important. The activities of this molybdenum, and thus it is also present in the nitrate reductase enzyme. That is important. And coming to the manganese. Manganese present in OEC, oxygen evolving complex, and plays an important role in splitting up of the water, photoionization of water, and also for the binding of ribosome subunits and DNA and RNA synthesis. That is the manganese. Iron already discussed, and the sulfur already discussed. Zinc also discussed. That is about the various activities or the role of the elements in the plants. So these elements are required to certain concentration. Below that concentration, the concentration at which it is required for the plant's growth and development is called critical concentration. Critical concentration, below which the growth in the plants is retarded, and we can observe this by the morphological changes. Morphological changes indicates the deficiency of the elements, and especially the KCN, that is potassium, calcium and nitrogen deficiency we can observe in the leaves, and sulfur and the calcium deficiencies also can be noticed. There are some morphological differences. Say for example, loss of chlorophyll is called chlorosis. Chlorosis is because of the deficiency of the nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc and molybdenum. And necrosis is the death of tissues. It is chiefly because of the deficiency of calcium, magnesium, copper, potassium, especially the nitrogen, potassium and the sulfur. These things, they inhibit nitrogen, potassium, sulfur, very important, inhibit the cell division, that is also very important. And coming to the toxicity. If any element is less than 10% in dry weight, it becomes toxic. Less than 10% it becomes. Sometimes more than 10% also inhibits the activities of other elements. For example, manganese, when it is in excess, it inhibits the action of the calcium. That is very important. And the manganese competes with the magnesium and iron and inhibits the calcium in the shoot apex. And as we know, in the absorption apoplast, in the previous chapter I explained, apoplast so the cell wall, intercellular spaces, and the passage cell, and into the xylem, and it is the passive, which does not require energy, and at the same time it is very fast. Simplast is a slow process as it enters through the root hair and into the cells and the cortical parenchymatous cells, and pass through the plasmodesmata to reach passage cell and finally the xylem, and it takes place at the expense of energy. Coming to the nitrogen cycle. Atmosphere consists of 78 to 80% of nitrogen. This nitrogen reaches the soil in two ways. That is biological fixation and high energy fixation. There are two ways of biological fixation: the symbiotic fixation and non-symbiotic fixation. Symbiotic fixation The root nodules of leguminous plants contain rhizobium bacteria, and then this rhizobium bacteria fix atmospheric nitrogen symbiotically. This is very important. Symbiotically, it fixes the atmospheric nitrogen. And non-leguminous plants like ulnus also contain Frankia, a bacteria which fix atmospheric nitrogen symbiotically. At first, and then these the rhizobium also present in the Frankia as well. Frankia present in ulnus, a non-leguminous plant, whereas the rhizobium present in not only in the different plants like alfalfa, sweet pea, sweet clover leaf model of tRNA, sweet clover, and lentils, broad pea, garden pea, etc. And how they invade into the roots, that's very important, and given in the textbook, that is also very important. At first, the rhizobium or Frankia present in the soil. They make their way into the plant through the root hair, and it makes the rhizobium makes the curling of the root hair. With the curling of the root hair, they enter into the cortex region where they divide more, and the cortical cells. In the cortical cells, nodule formation takes place after the infection of this rhizobium. That is very important, and the diagrammatic representation also given. Kindly go through it. That is very important, how the nodule formation. In this region, nitrogenase and the leg hemoglobin is present. As the leg hemoglobin is present, in the nodule, you can see the pink color. It is because of leg hemoglobin. So this is about the biological fixation. That is the root nodules of symbiotic fixation. That is, root nodules of leguminous plants contain rhizobium bacteria which fix atmospheric nitrogen symbiotically. And whereas there are certain free living soil bacteria like Astobacter, Azospiridium, Bergenicia, Rhodospiridium, Clostridium, which is anaerobic bacteria, they fix atmospheric nitrogen non-symbiotically. Anyhow, non-symbiotic and the symbiotic fixations together forms a biological in the presence of thunder and lightning. The nitrogen also dissolves in rainwater and reaches the soil in many ways. This nitrogen, which has reached the soil in many ways, will be converted into ammonia. This is very important. Conversion of the nitrogen into ammonia is called ammonification. This takes place because of enzyme called nitrogenase. That is very important, not because of bacteria as we have previously studied in many books. It is given not because of bacteria but nitrogenase. This is very important. So that is N2 plus 8H plus 16 ATP. 16 ATP gives 2 NH3 and the ADP, 16 ADP and 2H plus. So that reaction takes place in the presence of the nitrogenase, and this is the first stable compound produced during the nitrogen cycle, that is the ammonia. This ammonia is converted into the nitrite by nitrosomonas, nitrosocystis. This converts this ammonia into nitrite. Nitrite are converted into nitrates by nitrobacter. So this nitrates, plants absorb in the form of nitrates, and the remaining nitrates will be converted into free elemental nitrogen by denitrifying bacteria like Pseudomonas thiobacillus. Thus, the movement of nitrogen always is cyclic. So once again briefly, biological fixation, symbiotic fixation, non-symbiotic fixation, high energy fixation, and then converting the free elemental nitrogen into ammonia by nitrogenase, ammonia into nitrate, and again into free elemental nitrogen, called nitrogen cycle. This nitrogen, which is absorbed like this, and then here are the equations also given. Then ammonia 2NH3 plus 3O2 gives 2NO2 and water and 2H that is nitrite, and 2NO2 plus O2 gives 2NO3 nitrate. So that is nitrobacter. All these bacteria, they are the chemosynthetic bacteria, as you have studied in the chapter 2 itself. Chapter 2 in the first PC, we have studied all these things about chemosynthetic bacteria. So by breaking down inorganic salts, they synthesize the energy obtained from the inorganic salts like nitrate to nitrate. Such bacteria are called chemosynthetic bacteria. So what is the fate of this nitrogen? This nitrogen, which has entered in the form of nitrate, will be converted into amino acids. And formation of amino acids, there are two methods: reductive ammunition and transamination. Reductive ammunition, for example, alpha ketoglutarate acid plus ammonia plus NADP H gives glutamate plus water and NADP. That takes place in the presence of an enzyme, glutamate dehydrogenase, and this called reductive ammination, as ammonia will be added to the alpha ketoglutarate acid to give glutamate. And one acceptor, another one is donor. Then the acceptor accepts the ammonia group, that is called transamination. This takes place in the presence of transaminase, and the nitrogen is present in the form of amides like glutamine, aspargine. Glutamine and aspargine are formed because of the addition of ammonia at the OH group of the acid, and they contain more nitrogen, and they are transported, these amides are transported through the xylem. Similarly, in soybean, there are certain compounds called urics. They contain more nitrogen than carbon, and this is about the explanation of the chapter number 12, mineral nutrition. Very important chapter. Please read sentence by sentence and try to score more marks in your NEET examinations. Thank you. Please subscribe.